Welcome back, everyone, to the deep dive. You know how sometimes when you're trying to remember something, events from the same day, they just kind of clump together in your mind, like they're all part of one memory almost?
Yeah, for sure.
But then something that happened a couple days before can feel like ages ago. Like it's so much further away. It's a weird phenomenon, right?
Absolutely. It's like our brains have this built-in time warping mechanism or something.
Well, get this. There's some really fascinating new research coming out of Ohio State University. They actually published this study in Nature Neuroscience. And it digs into the actual physical mechanisms for why this happens, why our brains link those memories together.
It's super cool. They used mice to study how the brain connects these experiences, and it provides a lot of insights into how memory works, really at a fundamental level.
So in this deep dive today, we're really going to try to get down to the nitty-gritty of how these connections are actually created. So we know that we do link memories together, right? Like that's pretty clear.
Right.
But we want to figure out how that process happens, like in the nuts and bolts of the brain. What's going on at the neurological level? So first, let's just start with that common experience. So why do events that happen around the same time often feel so connected in our minds? What's the basis for that feeling of connectedness?
Well, it's something that everyone experiences, I think. It's pretty universal. And, for a while now, researchers have been trying to pinpoint the exact neurological processes that create this linkage.
Yeah.
I mean, we've known that when things happen close together in time, there's often some overlap in brain activity patterns, but the specific mechanisms that actually tie those memories together have been kind of a mystery. Like, what makes those memories feel like they belong together in our minds, right?
Right, right. So traditionally, when scientists and just, people in general have thought about memory, they've focused a lot on the neuron cell body. Like that's kind of the central hub of the neuron, right? But this new research really emphasizes the role of something called dendrites, which are kind of like branches extending out from the cell body. Can you give us a little refresher on what exactly dendrites are and why this shift in focus is so important?
Sure. So you can think of a neuron like a tree, okay?
Okay.
The cell body is like the main trunk of the tree, and the dendrites are those branches reaching out in all the directions.
I see.
And those dendrites are where the neuron receives signals from other neurons, so it's really all about communication. And for a long time, the thinking was that the cell body, the trunk, did most of the processing and storing of information. Like it was the boss, basically.
Right.
But now, thanks to this study and a lot of other research too, we're realizing that dendrites are much more active. They're not just passively relaying information. They're actually doing a lot of complex calculations on their own, like little mini brains almost.
Wow. So that analogy of the tree really helps kind of visualize that. And one of the researchers in the study, Mega Segal, she actually described dendrites as like tiny computers inside the neuron.
Yeah, a perfect description. Each dendritic branch can essentially process information independently. And that means that a single neuron can handle much more diverse information.
Mhm.
And what's exciting about this study is that it's suggesting that these tiny computers are playing a central role in linking those memories that occur close in time.
So to test this idea, they actually conducted experiments with mice. Can you walk us through how they set up the experiment and what they saw?
Absolutely. So what they did was they took these mice and exposed them to two different environments within a few hours.
Okay.
And then in just one of those environments, they gave the mice a small electric shock, just enough to make them a little uncomfortable, right?
Mhm.
Not painful or anything. But what they found was fascinating. The mice started showing fear in both environments, even though they only got the shock in one. So even in that environment where nothing bad happened, the mice were still acting scared.
So basically, the mice's brains were associating the two places, even though the negative experience, the shock, only happened in one. So it's almost like their brains were saying, hey, these two things happen close together, so they must be connected somehow.
Exactly. Their brains were basically linking those two experiences because of their close timing. It's like they were forming a single memory that included both places.
So that's a pretty clear demonstration, like behaviorally, that memories form close together. They do get linked. But then how did they actually figure out what was going on in the brain, like specifically?
Right. So they focused their attention on a specific area of the brain called the retrosplenial cortex, or RSC.
Okay.
And that area, it's known to be super important for spatial memory and contextual memory. So remembering where you are, the surrounding circumstances of an event. And importantly, the RSC is thought to be involved in linking events together in a sequence. So it made sense to look there.
So they basically zoomed in on the RSC, and they watched what was happening inside the neurons as the mice were forming these memories.
Yep, and they found something really cool. When the mice were forming these linked memories, they saw that the same groups of neurons in the RSC, even specific branches of those neurons, were consistently firing. So it wasn't just a general increase in activity in the whole area, but a very specific pattern within individual cells.
And then it gets even more interesting. They went even deeper, looking at those tiny protrusions on the dendrites, the things called dendritic spines. What were they seeing at that level?
Right, so these dendritic spines are like little knobs on the dendrite branches.
Okay.
They're the points where neurons connect and communicate with each other. And the researchers observed that whenever a new memory was formed, new clusters of these dendritic spines would pop up. And these clusters, they essentially strengthen the connections between neurons. It's kind of like adding more lanes to a highway, makes the information flow faster and easier.
That's a great way to put it. So more connections, stronger memory. But what was the key finding about these clusters? Did they behave differently when the memories were formed close together?
Yes, that's a crucial point. Those dendritic spine clusters that formed after the first memory were actually more likely to attract even more spines when the second memory was formed right after. It's like the brain was saying, hey, we just built some new connections here. Let's keep building in this area.
So it's like a snowball effect.
Yeah.
Like once the memory gets encoded in a particular spot on the dendrite, it's easier for related memories to kind of piggyback on that spot.
Exactly. It creates a kind of prime zone on the dendrite. It makes it more likely that new connections related to a nearby event will form in the same neighborhood. And the researchers actually said in the study that it's almost like memories are stored in dendritic compartments. When one memory forms, the affected dendrites are primed to capture new information arriving within the next few hours, linking memories formed close in time.
Okay, that's mind-blowing. So basically, your brain is physically tagging events that happen close together, and it's doing this by setting up shop in the same tiny neighborhood in your neurons. It's like putting all the related files in the same folder on your computer. And that co-location, that physical proximity of those memories on the same dendritic branches, it explains why, things like your morning routine and your commute, even though they're different things, they feel like one smooth block of time in your memory.
It really highlights how efficient the brain is at organizing information.
Totally. But they didn't stop there. They took it even further with this technique called optogenetics. Can you quickly explain what optogenetics is, just for our listeners who might not be familiar?
Sure. So optogenetics is this really cool tool that allows scientists to basically control neurons with light.
Okay.
They insert special genes into neurons that make them sensitive to light. So then by shining specific wavelengths of light onto these neurons, they can turn them on or off, like really precisely.
Wow. So like a light switch for neurons?
Pretty much. And in the study, they used optogenetics to activate those specific dendritic segments that were active during the first memory. What they wanted to see was whether they could artificially link two memories just by reactivating those dendritic segments.
Okay, so what happened?
Well, here's where it gets super interesting. Just by reactivating those specific dendrites with light, they could create a link between two totally unrelated memories in the mice. Even though the events didn't happen close together in time, they could artificially trigger that priming effect.
Wow, so they were basically like hacking into the memory system. Just by shining a light on these specific dendritic branches, they could make the mice's brains think that two separate events were connected.
Exactly. And that's a huge piece of evidence that those localized changes in the dendrites aren't just, a side effect of memory linking. They're actually a key driver of the whole process. So it really underscores how powerful dendrites are when it comes to organizing and connecting our experiences.
So this is really changing how we think about memory in general. It's not like we're just passively recording isolated events in our brains. We're actually building this huge interconnected web of experiences.
And Mega Segal actually said, the idea is that we don't form memories in isolation. You don't form a single memory. You use that memory, make a framework of memories, and then pull from that framework when you need to make adaptive decisions.
I love that. It's like we're constantly weaving a tapestry of memories.
Right. And this dendritic linking seems to be a crucial part of how we build and maintain that tapestry. It lets us organize our experiences in a way that makes sense, both in terms of time and context, and it helps us access those memories more effectively.
And this dendritic mechanism, it just seems so efficient, right? I mean, it makes sense that memories that are closer in time would be more tightly linked. They're more likely to be relevant to each other, or they might happen in a similar environment, you
Yeah.
know? So this process allows our brains to really streamline how we store and retrieve information.
Totally. It's all about creating the most efficient pathways for accessing the information we need. So by linking those temporally close events at the dendritic level, our brains can retrieve related information faster and even make better predictions about what might happen next based on past experiences.
And beyond just the science of memory, this research also has some really promising implications for therapy, especially for memory-related diseases like Alzheimer's.
Absolutely. If we can understand exactly how memories are encoded and linked at this level, it could lead to new ways to help people whose memory isn't working properly.
Right.
Mega Segal even mentioned that our work not only expands our understanding of how memories are formed, but also suggests exciting new possibilities for manipulating higher-order memory processes. This could have implications for developing therapies for memory-related conditions such as Alzheimer's disease.
So essentially, by understanding the mechanisms of these tiny computers inside our neurons, we might be able to find ways to help people with memory loss.
Exactly. The more we learn about the details of memory, the better equipped we'll be to tackle those really challenging diseases. And this study is a great reminder that we need to keep looking deeper.
Hmm.
It's not just about the neuron's main body, but also about the amazing complexity of those dendrites and what they're doing.
So for our listeners, let's wrap things up with the key takeaways from all this.
Yeah.
What are the main points you want everyone to walk away with?
So, here are the big takeaways. First, memories that are formed close in time, they're not just associated in our minds, they're actually physically linked together within the dendrites of our neurons. It's a physical connection.
That's huge.
It is. And second, this linking, it happens in specific branches of those dendrites, and it's mainly concentrated in brain areas like the retrosplenial cortex, which deals with spatial and contextual information.
Okay.
And third, those dendritic spine clusters, those little connection points, they're the stars of the show. They really help to create these links by acting like anchors for new memories that are formed close to existing ones.
So those tiny little things are like the glue that holds our memories together.
Exactly. They help create that framework of memories that we rely on so much. And finally, what's really exciting is that all this knowledge about how our brains organize memory at this super detailed level, it could pave the way for new therapies for memory disorders.
It's incredible to think that those microscopic branches on our brain cells are so crucial for how we experience and remember our lives. This discovery that our brains physically link memories that happen close in time, right there at the dendritic level, it's just a huge step forward in understanding how memory works.
It really is, and it makes you wonder, how might we use this knowledge to our advantage? Could we improve our learning or memory by intentionally grouping related tasks together, by taking advantage of this dendritic linking?
That's a great question.
And what other effects might this physical linking have on things like our perception of time or even how we experience the world?
Those are some really deep questions for everyone to ponder. And we've only just scratched the surface of the fascinating and complex world of memory. So if you're interested in learning more about this study, we definitely encourage you to check out the original research in Nature Neuroscience.
It's a great read.
Thanks for joining us for this deep dive, everyone. We'll see you next time.